Assalamu alaikum and a very good evening, everyone. I hope you have enjoyed yesterday's session. I am Sabah Sheikh, Assistant Professor at the School of Pharmacy on the behalf of Anjuman-I-Islam Kalseka Technical Campus, New Panvel, extend my warm welcome to all dignitaries, resource person, faculty member, participant from different colleges across the India to day 2 of 1 week AICTE-ISTE sponsored refresher program on multidisciplinary and translational drug development. To start with, as a tradition of Anjuman Islam, I request Professor Salman Beg to recite and translate few verses of Holy Quran.
أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم وعباد الرحمن الذين يمشون على الأرض هولا وإذا خاطبهم الجاهلون قالوا سلاما صدق الله العظيم. And the servant of the gracious Allah are those who walk on the earth in a dignified manner, and when the ignorant address them, they say peace. Al-Furqan.
Thank,
Sadaqallahul Azim.
thank you, Professor Salman. These verses from chapter Al Furqan of Holy Quran are worth implementing in our life. Yesterday, we had two very excellent and engaging sessions. The first session was conducted by Dr. Muhammad Farooq Sheikh, Senior Lecturer, Jeffrey Che, School of Medicine and Health Sciences, Monash University, Malaysia, on innovative disease model. He discussed about translational research, drug development timeline, animal model in drug discovery, case study of zebrafish for anticonvulsant screening, zebrafish in personalized epilepsy care, and he also shared some success story of zebrafish modeling. The second session of the day one was conducted by Dr. Abdul Basil, Assistant Research Professor, College of Pharmacy and Pharmaceutical Sciences, Washington State University, on drug transporter, case for interindividual variability in drug exposure. During his session, he briefed about drug transporter, its classification, tissue localization, clinical significance, and its application in drug development. Going ahead, today we have a very interesting session on computer-aided drug design. For this session, we have with us eminent resource person Dr. Nafis Ahmed, and it's my privilege to introduce Dr. Nafis Ahmed. Dr. Nafis is working as senior lecturer in medicinal chemistry at School of Pharmacy, Monash University, Malaysia. He obtained MS and PhD in natural products chemistry, medicinal chemistry from Naipur, Mohali, India. He worked on total synthesis of natural products, synthesis of new chemical analog for discovery of new anti-HIV agent based on natural product templates. His areas of expertise are computational based rational design of new drug candidate for HIV-1, dengue and other therapeutics. He has written many book chapters and published many articles in national and international journals. With this brief introduction, I extend my warm welcome to Dr. Nafis Ahmed and request him to start with the session. Dr. Nafis Ahmed, sir, it is over to you. Thank you, sir.
Thank you very much, Sabah Sheikh. Good evening to all of you and Assalamu alaikum. Let me share my screen to start with. First of all, I would like to thank Dr. Sharik and Dr. Sufyan on behalf of AIKTC for giving me opportunity to share some of my research experience here. So I'm going to talk about computer-aided drug design and optimization of molecules, which we are doing in my lab here at Monash University. So outline of my presentation, I will briefly talk about traditional drug discovery process and modern drug discovery process. And in details, I will discuss about work which we have done in my lab about anti-cancer drug development as well as anti-HIV drug development. And at the last, I will conclude my presentation. So to start with traditional drug discovery process, as all of you know, that there are different sources of drug. Mainly, we can categorize into two: small molecule compounds as well as large molecule compounds. So small molecule compounds, they may be nature products obtained from the nature, or they may be synthetic molecules. And if you compare these two molecules, synthetic medicinal molecules are much in percentage compared to nature products. We also have biologicals, especially proteins obtained from the nature or synthesized via recombinant DNA technology. As we know, these are the few examples of sources of drugs we can obtain either from nature, including plant, animals, marine sources, but majority of the compounds nowadays is from the synthetic medicinal chemistry. We can say that 90% of the molecules which are being developed and come into the market are from synthetic molecules. So if you look traditionally how drug discovery process take place earlier, okay, based on the molecule we have in our hand, depending on various hypotheses, that may be physiological hypothesis, it may be molecular hypothesis based on earlier mechanism of action of known drugs, or based on the chemical similarity with the known drugs, we can screen molecules against various targets. We have various biochemical assays or cellular assays, and based on that, you can say preliminary screening, we get some initial hit compounds. Once we get initial hit compounds, then we do in more detail about various biological evaluation. For example, we look into mechanism of action, dose response curve. We look into in vitro cellular work and also in vivo work to optimize pharmacokinetic and pharmacodynamic properties. Once we get some lead molecules, we can further do optimization in terms of potency, selectivity, physical parameters can be changed. And once all these are done, then finally we can do clinical trials in the human beings, and ultimately we get some molecules in our hand for ready to be tested for clinical trials as well as in the market. So if we can see from this chart, normally drug discovery process takes almost 15 years, 10 to 15 years, and first 5 to 6 years is mainly in the drug discovery process, which we call as preclinical testing. And once molecule pass from preclinical testing, then only we perform phase 1, phase 2, and phase 3 trials so that molecule can be launched into the market. So in terms of cost, if you see, we can say that a pharmaceutical company spend almost 500 to 800 US million per molecule, and success rate is very, very low. Out of 5,000 to 10,000 compounds, maybe possibility is that only one molecule will clear all the preclinical and clinical settings and will be launched into the market. So for chemical drugs, we can say that time duration is almost 15 years, and for biological drug, it is almost 10 years. So since traditional way of drug discovery is very tedious, time consuming, nowadays almost all the pharmaceutical company focus on modern drug discovery process, or that is also called as computer aided drug design. So it is one of the strategy for us to, for especially for the pharmaceutical company to manage with that time and money. So what basically happens in modern discovery process. Since traditional way of drug discovery is very costly, time consuming with very low success rate, and nowadays we have advancement in biomedical sciences where we have the structure of proteins, enzymes, and various targets using various protein, for example, X-ray crystallography. We now know the structure of targets. So with that in hand, we already know what is the target for most of the drugs. Also, nowadays we know much better about the molecular mechanism behind various diseases and disorders, what is wrong with a particular disease, what protein or what enzymes are hampered in a particular disease. So based on that, we know precisely about various diseases. So we know what play a role in a particular disease. So based on that, we can target a particular protein or part in the body so that we can treat that particular disease or disorder. And obviously, nowadays, there is increasing cost of experimental equipment and resources, which make drug discovery again costlier. And if you see, especially in the last 30 to 40 years with the computer revolution, nowadays we can say that there is very high speed revolution in the computers. The computers are becoming very low cost with very high power. There is a very high speed of software development. So with this, why not we can use computer for drug discovery process? That's why it is said that the speed with which computer sciences has progressed, if with the same speed automobile industry would have progressed, we can get a car or maybe Mercedes or maybe BMW just within 1 lakh. But we can see the speed with the with the automobile industry is progressing, the speed with computer sciences is progressing. There's a huge difference. So steps involved in drug discovery process, we know the structure of target. We can find out known agent or known drug which is acting on that particular target. Based on that, we can make a model to see how the binding is taking place with the help of X-ray crystallography, or in case X-ray crystals are not available, we can perform homology modeling. Then we can also search in the database for the new compounds based on structure of target. After that, we can dock a new molecules into the binding site of the target. We can predict the binding constant or activity. And once everything works well, we can synthesize new molecules for biological evaluation. So why it is possible nowadays? We can see there is very uh, high speed progress in the development of biomedical sciences. And nowadays we have a lot of proteins whose X-ray structures are known. So we can see that more than 150,000 proteins are available in the protein data bank. And the growth rate for addition is 100 to 200 new proteins per month. And we have various databases for small molecules. Basically, they are synthetic molecules, for example, NCI database, A molecules or ACD. And with that in hand, and along with computational advancement, we can use these together in order to fasten the drug discovery process. Various programs are available for this purpose. Here are the some list I have mentioned, but this is not inclusive. We have a number of softwares available nowadays from the different pharmaceutical companies. Some are paid while some are free. So normally in drug discovery process, we can use two strategy where we know the protein, means structure of protein, and based on that we can find the potential binding ligands which can bind to that particular protein, and we can search ligands from various chemical database. Another approach which is quite new, where we know the ligand, its biological activity is known. Now we can search various protein targets from the protein data bank so that we can find out potential application of that particular ligand for the treatment. For example, this new approach we can use for the treatment of COVID-19, where we know the structure of COVID protein, but we don't have any ligand. So we can use this approach, and nowadays pharmaceutical companies are using this particular approach for finding out new drug for the COVID-19. That is also called as repurposing of drug. So if you broadly classify various process which are involved in computer aided drug design, we can say that they are structure based or they may be ligand based, and further they can be categorized into ligand based docking or fragment based design. In case of ligand based, we can do ligand based virtual screening, QSAR, and pharmacophore modeling. And ultimately, all these processes are used in combination sometime in order to optimize lead molecules. So that was a brief about drug discovery process. Now I would like to share about our research work, which we have used this approach for the development of new anti-cancer molecule, basically for the treatment of breast cancer. So about breast cancer, according to Globocan, it is one of the most common cancer in women, and it accounts for 25% of all the cancers. So you can say it is 1/4 of the cancer amongst all. And particularly in Malaysia, according to Malaysian National Center Registry report, it was found that age standard rate for the breast cancer is about 31 per 100,000 of women. So that is quite high in Malaysia. And if you see the death rate, breast cancer is also one of the cause of cancer death among the women worldwide. We can categorize breast cancer into different classes based on which proteins are expressed in a particular breast cancer. For example, we have luminal A, luminal B, we have HER2, we have basal-like cancer, and all those. At the moment, available treatments are either hormonal therapy, where we can use SERM or SERD modulators. For example, is tamoxifen. We can use chemotherapy, where the synthetic molecules are used. Radiation is another option, and ultimately surgery is done. In case of hormonal therapy, it was found that 50% of the cases were relapse, those who are on the tamoxifen. So we can say that 50% chances are that cancer will relapse because of the tamoxifen resistance. So there is need for better drug which can be used for the treatment of breast cancer. So why we were particularly interested in G protein estrogen receptors. So this G protein estrogen receptor, it is a estrogen binding receptor. However, unlike the classical estrogen receptor, it involves non-genomic rapid signaling pathway. So it is not like a G protein coupled receptor or normal estrogen receptors. And it has been reported that this G protein estrogen receptor contribute to breast cancer proliferation, migration, as well as invasion into other parts. And high level of expression of these proteins or receptors have been observed, which lead to poor prognosis in the breast cancer. Also, there are proof about in vivo as well as in vitro, where there is link that these G protein estrogen receptor are overexpressed in the breast cancer. And one of the experiment is done where they have knocked down G protein estrogen receptor gene, and they found that absence of this particular gene prevents proliferation of TNBC, that is a triple negative breast cancer. So it clearly indicates that G protein estrogen receptors play important role in the breast cancer. So this is a structure of G protein estrogen receptor. So it belongs to GPCR. So its structure is basically same as G protein coupled receptor. It consists of 7 transmembrane domain. We have various molecules which are called as GPER1 modulators. So some are activators while others are inhibitors, and they have been used in various circumstances in treating cancer as well as other pathological disease. Here are the few examples. From the literature, we have selected these three molecules which are called as G1, G15, G36. And G1 was found to be a agonist at GPER1 receptor, while other two are antagonists. And they have very good affinity to these particular receptors. So based on that, though it is well known that GPER receptors are involved in progression of breast cancer, but there is no drug until now which can target GPER1. Okay, we said it that the another problem is there is no crystal structure available for this particular receptor. That's why there is little information available about its pharmacophore requirement in order to bind to this particular receptor. So based on that, we aimed at let's design a new ligand which can target this particular receptor, and we can form a pharmacophore study so that we can come up with the new molecules which can target this particular protein. So we have used in silico as well as in vitro approach for this particular study. So basically our aim was to develop a new molecule for treating cancer, especially in this case, a breast cancer. So we have objectives that we can design a new G protein estrogen receptors using various computational approach. Then we can synthesize molecules based on our in silico results. Then we can evaluate for anti-cancer activity, and based on IC50 values, we can again perform a pharmacophoric study so that that will be helpful for future optimization and designing of new molecules. Okay, so this is the overall approach which we have used. So this work was done by one of my PhD student, Shafi. So what we have done, basically we have divided our work into three main category where we have done in silico work, synthesis work as well as biological work. And in silico virtual screening, we have done ligand-based screening where we have done ROX screening as well as ion screening. And in structure-based virtual screening, we have performed homology modeling, MD simulation as well as molecular docking. So we have used shape-based as well as structure-based virtual screening using G1 G1 ligand, which is a known agonist at GPER receptor. So coming to shape-based screening, what we have basically done, we have used ROC OpenEye software. So we have used G1 as a query model because it is well-known ligand which is capable of binding to GPER receptor. So various conformers were generated using Omega. Then we have used database which is called as e-molecule database consisting of almost 7 million compounds. So this database was used against this particular query model. We have used rocks, which is basically a rapid overlay of chemical structure. So it basically takes into consideration of uh, shape Tanimoto, which is about a shape of the molecules. So this particular parameter take into consideration of shape of the molecules and screen the database according to that particular shape. Another one is color Tanimoto, which takes into consideration of atom present in the molecules. And this Tanimoto combo is combination of color and shape. So we have used rocks which take into consideration of both of these parameters and screen the molecules. So from this database, we got a hit of almost 5,000 molecules, which were having a ROX TC of more than 1. After that, we have used another parameter which is called as ion or electrostatic potential similarity calculation. So it takes into consideration of electrostatic similarity in the molecules. And when we have used this one, it again screen out the molecules and we got a 500 hit. So this is G1. So it consists of 5 rings, 3 hydrogen bond acceptor, 1 hydrogen bond donor, and 1 hydrophobic portions. So this was used as a query model. In the virtual screening, we got a ROX TC of more than 1. So you can see here. So highest score was obtained as 1.81, while the lowest one was 1.2. Then about ion potential screening, what we have got here, the query, it consists of ET-COM score of 2, while highest one was having 1.68, this molecule from the database, while this molecule was having the lowest one with the COM score of 1.1. So the red area here, you can say it is a electronegative area. So basically on the part of molecule, the substituents which are negatively charged, for example, carboxylate ion, they are negatively charged, so they appear as a red, while positively charged substituent, for example, amines or guanidine compounds, they are positively charged, they appear as a blue. Now coming to structure-based virtual screening, since structure of G protein estrogen receptor is not known until now, so we perform homology modeling and we have used various software. We have used Discovery Studio as well as various online servers. When we have used Discovery Studio, these are the steps which are involved. So as you know, first of all, we need to find out the sequence of our target. In this particular case, we get the sequence of amino acids of this GPER1 from the UniProt. Its UniProt ID is Q99527. Once we get the sequence of amino acid, now we can use various templates. In this particular case, we have used BLAST and we found that 5 templates having similarity to our target. We have done alignment. We have prepared a homology model using a modeler, and the model generated have been assessed using various parameters, for example, rampage and Ramachandran plot, and ultimately model were refined with the help of mod refiner. So these are the 5 templates which we have used. Here you can see the sequence similarity. So we can say the highest one was 53 as well as 76, which was B2 adrenergic GPCR receptors. So using these 5 templates, we have generated various models. And here you can see after refinement, you can see the number of residues in the favor region. They are here. You can say almost like 96 to 98% of the proteins of the amino acids were in the favored region. We have used also various online servers, like for example, we have used iTester. So in that also we have used the same thing, but these softwares are online. So we just need to submit sequence of our protein of interest, and the server automatically generates various models. We have also used GPCR ModSim. These are particular special online server only for the G protein coupled receptors. So we have used this one, and it has used various templates for generating structure of GPER. So we have shortlisted 5 templates top templates which have been used by GPCR ModSim. Here you can see these are the DB, PDB ID of those proteins or templates here. So if you see, if you compare about iTaser, Swiss model, as well as ModSim, and using the multi template that is ModSim, here you can see we got here is more comprehensive, where we have used Discovery Studio, we have used iTaser, as well as we have used other servers. In this, you can see that after refinement, the number of amino acid residues in the favored region, we got maximum here in ModSim, which was almost 96%. So we have used this particular model for further docking. This is the model which we have finally chosen for molecular docking studies. Now about finding that binding site in that particular model. So it was predicted by using uh, CAST-P, which is called as Computed Atlas of Surface Topography of Protein Server. And it was found that it contains 40 catalytic sites. Here, one example of active site is given here. Active site 1, its area, volume, and interacting amino acids. So this is a particular active site 1. So like that, this software has predicted 40 various catalytic sites. Now coming to molecular docking studies. So we generate a target protein, GPER1 in this case. We prepare, optimize for docking. We use the active site. Then we have selection of ligands. In this particular case, we have used rocks to get 500 hit. So using those hit molecules, we have done molecular docking studies. In this particular case, we have used Fred because it was a large, huge molecule library. We can do optimization and ultimately we get some result and we can further uh, uh, study that. So when we have performed these molecular docking studies, here you can see we have few molecules like G1, G15, and G36, which are already well-known ligands. So these are the docking poses of these molecules with their interaction energy. So 500 compounds which were resulted from our base shape virtual screening were docked in this case. And for each ligands, we have generated 10 poses, and the pose with the lowest docking score were considered for analysis. And from that, we have chosen top 100 compounds that have docking score comparable to that of G1. So these are the few compounds which we got hit from the library. These are the 5 hit molecules. Then we have also done this binding interaction with the top scaffold. And these are the docking poses for these 5 hit molecules. So we can conclude that using 7 million compounds in database, we have performed shape-based as well as homology modeling, and we have ultimately come or narrow down to 10 hit compounds, 100 hit compounds, and from that we have chosen 5 topmost for consideration into synthesis. Now we have also performed validation of in silico studies by using retrospective validation. So I don't want to go much into detail about retrospective validation, but what we have done basically, we have used active database as well as decoys database. So active database consists of 15 active molecules, while from the uh, decoy finder, we have taken almost 500 decoys molecules, and various statistical evaluation were done. For example, rock score, AUC, as well as enrichment factors, and various virtual screening criteria were used, like color tonometer as well as shape as well as combo. And these are the results from the rock. So here we have calculated R square, and we found that there is a very good correlation, and our retrospective validation were justifiable. We have also done MD simulations to check the stability of homology model, and we have used 10 nanosecond, 0 picosecond, as well as 50 nanosecond, and we found that our model, which we have taken into docking, it was stable. Now coming to synthesis work. So this was the G1, which was query model, and this was the hit molecule we have obtained. So we have, if you can see here, there's very slight modification here where we have the uh, furan ring, hydrated furan ring, and here we have the pyran ring. So we try to synthesize these molecules, and here we have used acetophenone, and just simply in the microwave we have synthesized these two molecules. We have also performed some optimization so that we can synthesize a series of molecules. So for synthesis of 1,3-diphenyl urea as well as 1,3-diphenyl thiourea, various procedures have been reported earlier, but we have optimized these known procedures, and we found that when you treat isocyanate with aniline just in toluene at 0 degree, we just get a pure compound without any purification. So it was one pot synthesis, very simple, no purification was required with higher yield. So we have synthesized a series of compounds here. So I'm not going into details of that. So here basically I am showing you only those results which we have published. Then because of a limited supply of isocyanate, we have also uh, developed a alternative route. Here we have used ethyl chloroformate with aniline just at room temperature in DCM for 45 minutes. We get ethyl phenyl carbamate, which can couple with aniline to generate urea 1,3 urea derivative. So similarly, we have synthesized various molecules. So almost 50 to 80 molecules we have synthesized, and all were evaluated for culture work. So we have used 4 cell lines: SKBR3, MCF7, MDA, MB231, as well as MCF10A. We have performed first MTT assay to check cytotoxicity. So quite a standard protocol were used for this study. And it was found that various molecules were displayed higher cytotoxicity against SKBR3 cell line and also against other cell lines. So based on our result, we got very good promising result, which was because of uh, in silico design. And once we have obtained our biological IC50 values, now we want to study more in detail about binding interaction in the cell lines. So for that, we want to create a ligand binding assay with the help of fluorescent probe. So we have synthesized various fluorescent probe also, which can be helpful to find out mechanism of action in the cell line. And we have determined using flow cytometry. This work is actually not published. That's why I'm not sharing in detail about this. So to summarize this, okay, we have performed virtual screening and we have got some heat molecules, and based on those heat molecules we have synthesized around 50 to 80 different small molecules, and all were screened for biological evaluation. So we have used various cancer cell lines and results were promising. And to further do mechanistic study, we also perform binding ligand binding assay using fluorescent probe, and we have used flow cytometry for this purpose. Now coming to case study 2, which is about anti-HIV drug development. So just I can hold on. Meanwhile, I can drink water. Dr. Sharik, I am okay with the time or I am taking much time?
I think you're good on time. I think
Oh, okay, yeah.
we are, how long, how many more slides for this case study?
I think maybe another 30, 40 slides. So I will go much faster, maybe another 20 minutes.
That's fine. That should be fine.
Yeah.
Yes.
Okay. So this is another case study where one of my PhD student is working on anti-HIV compound. So as all of us know that HIV is one of the leading cause of death where millions of people are affected. And according to UNAIDS, it is about like 37 million people who have been infected worldwide. So nowadays we have a heart, which is a highly active antiretroviral retroviral therapy, which use a combination of drugs. But with the help of heart, we can say that AIDS is no longer a death sentence and it is a manageable. But the problem with heart is that we use a combination of drug which leads to drug resistance. In this slide, you can see these are life cycle of HIV virus, where various steps are like binding, then fusion to the human cells, then it undergo reverse transcription, translation, assembly of virus, and ultimately its release. And that is how it infects and multiply in the human body. So these life cycle indicate that we can target various sites in order to prevent progression of disease. And in the market we have drugs available which target this particular pathway. Like for example, we have reverse transcriptase inhibitors, we have integrase inhibitors, we have protease inhibitors, we have fusion inhibitors. So basically my idea was, okay, instead of using one single regimen, why not we can use a molecule which can target at multiple sites. So the problem for new drug development is because of various reasons. Like for example, HIV virus, it remain latent in the person's body, and at any time in the life cycle of its virus life cycle, it can be uh, it can multiply and the symptoms will appear. And another thing is that this particular HIV is also developing a fast resistance to known drugs which are used clinically. And when we use heart particularly, actually what we are doing, we are increasing drug load because we are using combination of drugs, and it leads to complex therapeutic regimen. It causes lack of compliance to the patient, and ultimately it leads to drug resistance because HIV is exposed to multiple multi-drug. So we propose that why not we can target life cycle of HIV at multiple sites simultaneously using one drug. So we can design one drug, for example, which can inhibit reverse transcriptase, which can inhibit integrase, which can inhibit protease also. So using single drug, multiple enzymes can be inhibited that will be more effective compared to heart. So that is called as a multi-targeting or multi-ligand, where we can use one drug targeting at multiple sites in the pathogenic pathway. And there are few successful story in that where people have tried to synthesize molecules with dual inhibitory activity. For example, here delaviridine, it is a reverse transcriptase inhibitor. So you can say that it is a mainly reverse transcriptase inhibitor and it has very low integrase inhibitory activity. But when it was substituted with a diketo substituent, actually it is quite effective to inhibit integrase enzyme as well. So we can play around the structure of molecules so that one molecule is enough to inhibit more than one enzyme. This is another story where we have some known drugs which are used clinically. For example, raltegravir, which is integrase inhibitor. It has been developed from etraviridine, which is a reverse transcriptase inhibitors. It was converted into these analogs, but it was found that it is inactive against integrase. Similarly, here we have etraviridine can be combined with eltiviragir. So we can use those substituents or pharmacophoric feature which is required for inhibiting that particular enzyme. And if you can combine into one molecule, let's see whether that combined molecule is effective against both enzyme or not. So looking briefly at the structure of reverse transcriptase, so we have looked into the literature and we found that the structure of reverse transcriptase, it is like a right hand. So you can see here, it consists of many sites. Red region, it indicates it is a polymerase active site, while yellow is the non-nucleoside reverse transcriptase site, where reverse transcriptase inhibitor bind here, while green is the ribonuclease active site and ribonuclease active inhibitors actually bind here. So you can say that all these polymerase active site, reverse transcriptase as well as ribonuclease active sites are very close to one another, and when one particular agent binding to one site, it can cause change in the shape of the protein and it can ultimately affect activity of other. So basically we can say, if you look into details, we can say that there are few hydrogen bond donor substituent present on the molecules which can form a hydrogen bond with the lysine 101 and 103 which is present on the active site. So especially RT active site, it consists of hydrophobic 4 and it consists of hydrogen bond forming region. So overall it is very flexible. So we have docked some molecules which is already reported in the literature, like rilpivirine. We have docked into 3-MeE, which is basically a reverse transcriptase, and we found that our molecular docking studies is actually same as reported here. So we can say on the right, on the left hand side here, actually there are few substituents here, lysine 101 and 103, which actually forms a hydrogen bond with the molecules. And on the other side, on the upper left, there is some molecules, some substituents or amino acids which can form hydrophobic interaction. In this case, it is a tyrosine 188 and tyrosine 181. Similarly, we looked at the structure of integrase enzyme, and integrase enzyme consists of three domains, and particular domain of interest is catalytic codomain CCD. It consists of three amino acid, aspartic, and glutamic acid, and it consists of a divalent magnesium ion. So two magnesium ion were present here, and they play very important role in activity of this particular enzyme. So if you can use benzyl hydrophobic moiety along with chelating trade, it can bind to magnesium ion and it can inhibit this particular enzyme. So here you can see in the red, which is a diketo moiety, it can chelate the magnesium ion. We have a benzyl moiety, which can form a hydrophobic interaction and it can inhibit integrase enzyme. So known drugs which are clinically used here, for example, altigravir, as well as dolutegravir, they all have these pharmacophoric features present. Like here you can see it has diketo moiety, which can chelate magnesium ion. They have benzoyl moiety to form a hydrophobic interaction with the tyrosine per se. Here also we have dolutegravir, and we found similar result as reported in the literature. So here it forms like a chelation of magnesium ion as well as hydrogen bonding. We have hydrophobic interaction on the top. So why we choose these two particular enzyme, that is reverse transcriptase and integrase? It is because of the various reason. We can say that these two enzymes have very similar structural similarity in terms of their topology. And if you see overall, the pharmacophoric feature required to inhibit this enzyme is quite similar. So we can integrate these pharmacophoric features into one molecules, and both enzymes can be inhibited at a time. So based on that, we have done some molecular docking studies. So we have come up with the pharmacophore model here. So we find that for reverse transcriptase inhibition, we have some sort of pharmacophore like that, where we have a aromatic core which is attached with another aromatic ring, but it must contain some hydrogen bond donor or acceptor so that it can interact with the acidic amino acids on the active binding site. On the same time, it has some non-specific part here on the left hand side, which can form maybe hydrophobic interaction with the receptor. So if you see here, actually on the right lower, we always have some substituent which can form a hydrogen bond here. And on the top left, we have aromatic ring which can be involved in hydrophobic interaction. So we have made various hypotheses based on this pharmacophoric feature, and we have designed various analogs based on quinoline, and we have docked these molecules to see whether our pharmacophoric model is working as per our information. So we have done ligand-based virtual screen in that case also, and one of the molecule we choose from the literature, which is this particular molecule, which is already patented in 2016, and we have used this as a query model. And in this case, again, we have used a molecule database to screen and come up with some interesting molecules. And we got some proposed as well as some molecules from the database, which are having quinolone moiety, and they are also hit in the database. So we choose almost like 1,000 hit. Then based on that, we have done some molecular docking studies on ribonuclease active site as well as reverse transcriptase site as well as integrase site, using known drugs which are used clinically, as well as our based on our hypothesis we made, our design molecules. And we found that our design molecule, for example, proposed structure 52, it was having quite good binding comparable to that of reference one. Here you can see the red arrow indicates where it can chelate magnesium ion. It was also able to bind well with the polymerase. We have docked with integrase also, and we also got a quite interesting result. So here you can see C-Docker interaction energy with the reference one, that is dolutegravir, and our molecule. So we can say it was quite comparable, and it shows that it has quite good affinity to the receptor. Then these are another structure which we have docked, and we got also very good binding affinity. So based on these docking studies, we have come up with the pharmacophore, which is required for binding to both enzyme, reverse transcriptase as well as integrase. And accordingly, we designed the molecules and we went for synthesis. So we have synthesized quinoline 2,4-diol using aniline with the diethyl malonate derivatives in the microwave, where we have synthesized substituted 2,4-dione quinoline derivatives. I'm not going into details. We have used substituted aniline so that we can come up with the range of molecules having diketo moiety at the same time benzoyl moiety, so that it have well interaction with the targets. This is another scheme where we have used thionyl chloride to convert one of the ester of diethyl malonate into chloride, and then we substituted one side of diethyl malonate, and later on then again we cyclize to get desired molecules. So we have made three scaffold, you can say, in the class of 4-quinolone based on our data screening. And accordingly, we have synthesized various molecules which belongs to these three scaffolds, which are amongst 4-quinolone derivatives. So these are the general scheme which we have used for synthesis of scaffold 1, A, B, as well as C. These are the few molecules which we have synthesized, and also we have synthesized some other molecules. Once we have synthesized these molecules, then we have done biological evaluation. So we have used HEK 293 cell lines for MTT as well as for MTT assay, and we have used reverse transcriptase and integrase kit. So it was basically kit-based essay. So still my student is some of the work he's doing, and we have got some very promising result. So once we complete our biological activity, then we will perform pharmacophoric studies to come up with the real pharmacophore, which can be further used for optimization of molecules. So these are the two. Another study about HIV, which one of my student is doing, and this also indicate that we have used computer-based approach, and it was quite successful. So to conclude my talk, we can say that computational based drug design is a promising approach. It is cost effective, less time consuming. We can use the knowledge of molecular biology as well as computational technology for this approach, and these all together actually increases chances for NCE. So better chances of getting a new molecule for optimization, less chances for failure. So that is some of the work which we have done and I have shared with you. So thank you very much for your attention. Sorry, I have taken a little bit longer time.
Thank you.
If you have any question.
Thank you, Dr. Nafis. I think it is very comprehensive. Thank you so much for showing some very interesting work that you've been doing at Monash in the areas of antiviral. There are a lot of questions in the chat box. One general question that I have essentially is, what happens typically is when you look at CAD, there are restrictions on the structures for proteins, and we're very focused on specific area. But what happens in biology, it becomes very complex. Essentially, you have receptors, you have interlinking with receptors, and there's not sometimes I've worked in the areas for HCC and other cancers essentially. So it's very complex essentially. And there are multiple pathways essentially. So in your experience, how does that, do you think that, CAT should be integrated more into with other, biological process to make it more relevant? Because now here we are seeing into a very isolated and a very closed area as such. So your thoughts on that essentially?
Yeah, so yeah, actually, basically, that is a, you can say, a drawback of computational work, because whenever we we consider all these docking studies, actually we consider a isolated compartment. But in reality, when drug acts or when the targets are available, obviously it is a biological system, and various proteins and other cellular uh, ions play role in the finally determination of activity. So one of the approach, actually people are so obviously that's why we say that this computational work, it's you can say its success rate is only 70% unless we really do in vivo work, because sometimes in silico as well as in vitro the system works, but in vivo, in the real system, maybe there are some interferences or some of the factors which hampered activity. So the real true picture will come into when we do in vivo studies.
I can
But
agree on
this
that.
give us at least some idea how we can proceed for further work.
Right.
Yeah.
So, just a follow-up question on that. With this whole, as you mentioned initially, there's a lot of exponential growth in the computational power that we have in this whole evolution of machine learning and artificial networks. Essentially, how do you see that field, enriching this whole drug discovery process in terms of intelligent decision making and intelligent target selection and stuff like that? Any thoughts on that?
Yeah, actually, I mentioned that especially when advancement in the computational chemistry is taking place, as well as artificial intelligence. So more we have the knowledge of the target, and we have the databases where the millions of synthetic molecules are present. So instead of earlier going to the blind screening and blind designing, we can use all these technology, and we can shorten our discovery process time. We can make uh, our research more focused by using all these areas. So definitely these artificial intelligence and all these technologies, they are very useful, and they are definitely leads to better success of NCE.
Excellent.
Yeah.
So there are a lot of questions out here in chat box. I don't know if you can read them there or not. One of them is our very own faculty, and I encourage other faculty. I see a lot of senior faculty members, many of them from medicinal chemistry background. If they want, they can either post their questions or they can ask us. If they raise their hand, they can unmute themselves. Either way, let's make it a little more interactive and make good use of Dr. Nafi's time. One of the questions is from our own faculty, Dr. Feroz Khan, in terms of which are different. Why he says is what is the need of using different softwares for the same study, and what at FDA, US FDA, what are the softwares that are acceptable in general?
Yeah. So actually, now if you can see that we have many softwares available nowadays in the market. Some softwares are free for academic purpose, while most of the softwares were paid softwares where you need to buy annual license. So actually, basically all these softwares work either on the physical chemistry principle, and they take into consideration of all the chemistry, especially organic chemistry aspect. So we have various softwares because various pharmaceutical companies or the organization or the universities, they have developed based on consideration of uh, various parameters. For example, some have taken into consideration of atomic charges, some have taken a consideration of only shape of the molecules, some have considered only uh, uh, similarly about stability of the molecules in the environment. So depending on the software, they take into consideration various aspect of the molecules. So in practice, what we do, we cannot rely only one particular software. As I told, still if you use multiple software, the reliability of the result is only 60 to 70%, meaning that whatever I am getting from the computational work, the same result I will get from in vitro and in vivo is only 60 to 70%. Because as we have discussed earlier, the real biological system was in a different way. But since these are based on the scientific uh, data and scientific background, we can say that these software, when we combine multiple, then we get more rigid data. It creates more confidence in us compared to only using one particular software.
Great.
That's why normally if you see in the papers, people use
Multiple
multiple
options.
software. So the result obtained from one particular software, they can validate by using another software. So if I use one software, I get one set of result. I get another software, I get another set of result, which are not matching. It means that there is something wrong, either in me or in my parameters. So I need to work around it. But suppose I am using multiple softwares and overall I am getting the same result, it we can say that there is more reliability on the calculation. That's why normally people use different softwares.
Professor Arul, also our faculty, he's asking about free softwares for CAD, essentially, especially looking at drug-drug interactions and known drugs with protein interactions. Are there any free softwares or
One of
something?
the free software is Autodoc. And most, if you see, like most of the people, those who don't have access to a licensed version, normally they use Autodoc and Autodoc Vina. So
Okay.
if you don't have any paid license, paid software, then obviously you can use Autodoc. But the problem with the free software is that you you must be expert in that. You need to learn a lot of things in that. And when you use a licensed version, actually many, for example, preparation of protein, preparation of ligand, many things is done by software itself. But in the free version, you need to actually do it. And that's why you must have expertise in that particular area.
Okay. Professor Jayashree is asking, she's asking about the HIV treatment. She says, as for retroviral regimen, usually NNRTIs are used rather than NRTI. Any specific reason or observations for NNRTI selection?
Yeah, basically, if you can see that mainly we use this NRTI in the clinics. The one of the reason is that because we know the role of reverse transcriptase in the HIV, and when we design any molecule which is based on NRTI, it is better in terms of its efficacy because it can easily bind to the, it mimics the nuclear bases, and the success rate is much better in NRTI compared to NNRTI. That's why most of the people normally focus on NRTI because of the similarity with the nuclear bases and well-known uh, mechanism of action. But still nowadays, if you can see in the literature, a lot of people use NNRTI because if you want to use NRTI, you have to stick to your chemical structure. You must have nuclear bases in that so that you will have a good affinity. But when you use NNRTI, you are free to use any scaffold because it is not targeting NRTI.
Excellent. Great. Any other questions? I don't see any questions in the chat box here. Anyone else would like?
Sharik sir, there are a couple of questions from YouTube live
Please,
session.
can you speak to
So
it, sir?
Yogesh Jagtap, he has asked one question that why you how can we compare LibDoc result with Glide? How we can
Yeah,
compare LibDoc result with Glide?
actually, if you want to directly compare these two, you cannot compare because LibDoc will give you different. It has a different parameter score. It gives a different parameters in terms of binding affinity, while other software will give different score. So what you can do based on that particular software, you can arrange the affinity of the molecules and then you can compare. So generally, you will find a similar trend. But if you want to directly compare the value of these two, actually uh, it is not a good way to compare directly the values. But you need to see the general trend, affinity values in both the in different softwares. So in general, they have the same trend.
Okay, all right. Uh, I think one more question from our one of the YouTube viewer. How you develop pharmacophore?
Pharmacophore for HIV. Normally,
No, it
for
is
the
general question. Yes.
if it is a general question that then in order to develop a pharmacophore, you must have some molecules with known activity, either IC50 values or at least EC50 values. When you have a set of molecules with the known biological activity, based on that, you can develop a pharmacophore. So you have a highly active molecule, you have intermediate active molecule, and you also have some inactive molecules. So system takes into all these three parameters, these three molecules, and based on that, then it creates pharmacophore for that given set of structures.
Okay, I have done with YouTube live questions.
Uh, Dr. Nikhat is asking essentially, can we get 100% in vivo and in vitro results with AutoDoc?
Never. As I mentioned that whatever we are getting from in silico, it is only 60 to 70% reliable. And when you do in vitro, obviously sometimes you get very good result in vitro, but when you perform in vivo, sometimes molecule completely fail. So
Absolutely.
even for in vivo to in vitro, we cannot compare. And when we compare in silico, in vivo, in vitro, uh, we cannot say that there is a guarantee that you will get the same trend. But
Absolutely. I mean, the,
chances are higher that when you do proper approach using in silico, then go to in vitro to in vivo, there are higher chances of activity.
great. Thank you, sir. I don't think there are any other questions here. If there are any other further questions, you can directly email us and we can forward it to Dr. Nafis. Um, I think it is over to you, Professor Sabah.
Yes, sir. Thank you, sir. I would like to thank Dr. Nafis, sir, for comprehensively answering all the questions asked by our participant. Thank you, sir. Thank you once again.
Thank you very much, all of you, and
Uh,
especially thank you, Dr. Sharik and Dr.
Thank you.
Sufyan,
Thank
for
you so much for giving
organizing
us the
this.
time, essentially.
Yes.
And so, before we close down here, I think there is a link for the feedback. And also, just to remind you, tomorrow we'll be having a very interesting speaker, Dr. Manasi Pimple, who will be coming from Florida, and she'll be talking about very interesting area of pharmaco, mostly into biomarkers and mostly looking into the areas of precision medicine as such, in terms of using biomarkers as a tool for patient selection, as a prognostic marker. So she has a lot of interesting data that she'll be sharing with. So hopefully, all as we've joined today, tomorrow also we'll join in for another exciting session for this FTP. So I think Professor Sabah, are we ready to close?
Yes, sir. So just